Yeah, so thank you so much for the invitation to participate in this conference. And I'm really sorry that I can't be there in person, actually. First of all, I think it looks like a great location, but also I think you really get much more of a feel for the conference when you're there in person and the people who are there. And I guess I really would have liked the opportunity to mingle with some of you. That's really the thing that you're lacking when you do it all online, I think. But nonetheless, of course, it's great to be able to share some of our work here with you and to participate in the discussions today, at least. Okay, so just to start off, I'd like to give you a little bit of an overview of what we actually do in my group. So we combine EPR spectroscopy, electrochemistry, and biochemistry to unravel the mechanisms of some pretty complex redox active enzymes. And numerous enzymes rely on redox reactions and are involved in quite essential biological processes, such as in respiration and also in photosynthesis. And luckily for us, unpaired electrons are often located in quite mechanistically key locations. So really what we are exploiting is what can be both considered a limitation and a major advantage of EPR spectroscopy. And that is the fact that the technique is really blind to everything but unpaired electrons and of course surrounding nuclei. So we can look at really quite huge proteins. So in the case of mammalian complex 1, so that's actually the bacterial one that I'm showing here, but the mammalian one is 1 megadalton. So in the first part of my talk, I'd like to talk about the work that we've been doing with Guy Hanke at Queen Mary University on the photosynthetic complex 1. And indeed, this builds on a lot of the methodology that we have developed for the respiratory homologue, which is much better understood. And in the second part of my talk, I'd like to talk about some method development that we've been doing in the group to advance ultimately our understanding of these redox based catalysis, especially in biology, although I think it is more generally applicable as well. And last but not least, as Chris already mentioned, I'm on a mission to build up a center for post-EPR at Imperial, and we actually have a launch event coming up soon. So if you do happen to be interested in EPR spectroscopy, then please do join us. It will actually be a hybrid event, both in person, but also with the opportunity to join online if that's better for you. So with that, let me give a brief outline of my talk. So this has really all been driven by Catherine for the first part, so I'll mention that shortly. But the first part will be about the photosynthetic complex 1, as I mentioned. So I'll give a bit of context and comparison to respiratory enzyme, and then I'll tell you about the characterization of these iron sulfur cluster centers in this enzyme, their assignment to the structure, and their properties, which is really what we are interested in to understand the function of this enzyme. And then the second part will be about film electrochemical EPR, and I hope to convince you why we need a new method and what we've done in terms of demonstrating proof of concept and a little bit of an outlook as to where we are with this right now. So during the light reactions of photosynthesis, electrons are extracted from water, of course, and reduce the plastoquinone pool here, which is coupled to proton pumping across the thylakoid membranes, as well as to the reduction of ferredoxin. And this in turn, of course, results in the production of NADPH for carbon fixation. So this is the so-called linear electron flow. But what can also happen is that the electrons go back into the plastoquinone pool here through photosynthetic complex 1. And formerly, this was actually known as the NADH-like complex, but I think it's really now quite well accepted or renamed to photosynthetic complex 1 because NADH-like complex is really a bit of a misnomer given that the electron donor is not actually NADH. And so although there are some similarities with the NADH ubiquinone oxidoreductase in respiration, which is where this came from in the first instance. So what happens here when we put electrons back into photosynthetic complex 1 is that we get an increased proton motive force, which leads to increased ATP synthesis relative to NADPH production, and that can really be very beneficial. So for example, for the cyanobacteria that we are looking at, this can be very useful for them under stress and enable them to survive under low carbon dioxide concentrations. So here's a bit of a comparison with the respiratory enzyme shown on the right-hand side here. That's a bacterial structure here. And you can see there's really quite a lot of structural similarity. And the most obvious difference is that the photosynthetic enzyme is missing a large chunk of the hydrophilic domain here of this enzyme. And of course, it's only got 3 iron sulfur clusters as opposed to this really long chain of iron sulfur clusters that we have in the respiratory enzyme. And much unlike the respiratory one, there's really hardly any biophysical studies of this enzyme, or actually there weren't any when we first embarked on the project in photosynthetic complex 1. So we have very little idea in terms of the bioenergetics of how this enzyme actually works. And that includes not knowing what the reduction potentials are of these clusters. So this is really where we were and what we set out to do. So can we actually begin by determining the magnetic parameters of the 3 iron sulfur clusters, because without that, we can't really go anywhere from there with more complex analysis with EPR. So that's really the starting point. And can we actually assign them to the structure once we have the EPR signature? And can we then learn something about their properties? But first things first, the big task is, of course, to isolate the enzyme. And this is quite difficult and time consuming. And really all the credit here goes to Catherine and Guy, and also Anna, who is the postdoc that Guy and I first had with some seed funding to get things started. And we chose to work with 2 cyanobacterial species. So the Synicocystis here is genetically tractable, which is in particular useful for future work that we are now doing. And the Thermosynicocystis, Synechococcus elongatus, is actually the one that we then have a direct comparison with some structures that are now available. So cryo-EM structures that have been published since, which is really very useful. And of course, also when you work on 2 species, then it's very nice to be able to actually make some comparison and to see how conserved the properties are that you are finding. So to start with, Catherine recorded some continuous wave EPR spectra that I'm showing you here. And I realized that maybe not all the audience here is all too familiar with EPR spectroscopy. So maybe let me spend a little bit of time here explaining what is going on. So we actually have derivative spectra in continuous wave EPR, which is perhaps somewhat unusual. You'll see the subsequent pulse EPR spectra are perhaps the more what you're more used to, the absorption spectra. And mainly why we have derivative spectra is really ultimately due to signal to noise improvements that we can make this way. But in any case, you can see we have really quite broad line shapes and more than 1 G value. And this was already very nicely introduced by Melinda Dewar. And of course, that's because you are not averaging out all these all these molecular orientations once you freeze your solutions. And indeed, we have to work with frozen solution spectra because otherwise our relaxation times are just too fast and we just won't see anything at room temperature, at least not with iron sulfur clusters, because their relaxation times are intrinsically really rather fast. So what we could do then by looking at the continuous wave spectra, we saw that we had 3 overlapping iron sulfur cluster spectra. And for now, you somewhat have to take my word for N0 really being there, because from the continuous wave EPR alone, there's absolutely no way that we could be certain about that. And the confidence really came from the subsequent post-EPR measurements. But what we could say right away is that N2 here matches very well with what we have in the respiratory homologue in terms of its G values. And therefore, we were quite confident to be able to assign this N2 signature here to the cluster that's adjacent to the plastoquinone binding site here. However, the same certainly can't be said for the N1 and N0 signals, which there's no direct equivalent with the respiratory enzyme. And so clearly that required some further investigations, and we need to actually define the G values for all the clusters and then decide also which cluster belongs to which iron sulfur cluster in the structure before we then go on to determine what their properties are. So in order to get these G values and G strains, so the magnetic signature, if you like, we played around a bit with relaxation time. So we looked at the T2 in the first instance. So by varying the tau, and this is the basic Hahn echo sequence here, and by varying tau, you can effectively measure T2. And you can see here that N2 has the longest T2, and we can start to effectively get it out quite nicely, this axial spectrum of N2, when we get to long tau values here in the sequence. Now we can play similar games with T1, so both by changing the shot repetition time, so that's the time, the delay between different pulse experiments, but also by using a refine method. So this is again the Hahn echo sequence here, and by putting a pi pulse in front of it with a variable time delay here, we can effectively filter out at least partially the contributions from different clusters, so that we can get more confidence on what the individual G values are, given that the clusters all overlap like that, which is really quite tricky. So this is quite a busy slide, but it's really just to show you that we were able to fit all the data quite nicely for these relaxation studies, and therefore we could come up with, I think, a trustworthy set of magnetic parameters for both sets of cyanobacterial species. I've just shown you the data here for T. elongatus, but we've got the equivalent of the Synechocystis in the paper as well. And really the take-home message from this is not only, of course, that we could get the magnetic parameters, which is important for the next step, but also that N0, so the broad one, has a very unusual combination of a relatively long T1 and a short T2, which might already be giving us some clues as to what its position is in the structure. But of course, we can't be certain at this point. So in comparison with the respiratory enzyme, as I already mentioned, we have a really good match with the N2 cluster here, but the N1 and N0 clusters don't really match any of the N-sulfur clusters in respiratory complex 1. And I should say that the cluster that's adjacent to N2 is actually not EPR visible in the respiratory complex 1 for reasons that we still don't fully understand actually. But even the next one along here, N4, doesn't match either N0 or N1 very well. So that didn't really help us any further. So which cluster is it adjacent to N2? And given that we have N2 assigned, this leaves us with two possible models, which I've just called A and B here. So either N1 at the furthest end or in the middle. And what we thought we could do is to use a pulse EPR technique called double electron resonance spectroscopy to effectively measure the distances between these paramagnets by exploiting the dipolar coupling between them. And really what we then need to do is to simulate these experimental Dia traces with the two possible models A and B, combining structural information with spectroscopic data. And that's actually quite a complex task. I will talk about it briefly shortly, because Dia with multiple N-sulfur clusters is really quite complex. But luckily, we could call on some methodology that I actually was driving quite a long time ago. So it was nice to dig this work out again that I did in my PhD, which was actually a first sort of taster for me into the complex 1 area. And what we solved there is, I guess, a controversy over the assignment of N4 and N5 in this really rather long relay of N-sulfur clusters in the respiratory enzyme. So let me explain a little bit more about Dia and how we apply that to multiple N-sulfur clusters. So the Dia experiment is designed to single out the dipolar interaction from all other spin Hamiltonian parameters. And this is achieved by having two different microwave frequencies. And in the absence of a pi pulse here at the second frequency, the primary echo here would just refocus at a time of 2 times tau 1. We actually use a dead time free experiment here. So that's why we have another pi pulse here to avoid the problems with the dead time that we would otherwise have. So what happens now if we have B spins? So they create a local magnetic field which affects the A spins. So when the pi pulse is applied and the B spins flip from the alpha to the beta state or vice versa, and this inverts the local magnetic field and changes the frequencies of the A spins, which therefore can't refocus anymore, and the echo is interrupted. So the pi pulse is shifted in time, and that causes a modulation of the refocused echo intensity, which is proportional to 1 over the distance cubed between the A and the B spins. So that is that if we measure this refocused echo intensity here as a function of time, then that's what produces the deer traces that you will see in the subsequent slides. And another, maybe just to mention here actually as well, that of course there's many different kinds of post-DPR experiments there that are designed to single out different interactions in your spin Hamiltonian, such as also the hyperfine interaction. But in this case, I'm just talking about the dipolar coupling. And luckily, in this case, we don't really have to worry about hyperfine interactions, so nearby nuclear spins, because that would make our picture even more complicated. But there's also some very nice work, of course, that you can do with hyperfine spectroscopy and so forth. So in the case of these really very highly anisotropic clusters, so spanning quite a large field range, as you can see here, we also have to take into account orientation selection. And that is because the pump and the detection pulses, so these observer and pump spins here, don't excite the entire Einthofer cluster spectrum. So what this means is that we have to obtain many different Deer spectra at different field positions in order to really get the distance, if you like, to obtain the complete peak pattern. So orientation selection can be an advantage, and indeed in our case it really is an advantage, because what you can see here is that the high field positions, we only have the N0 cluster contributing. So this should really help us in the assignment, in our task, to figure out what is the outermost cluster. And of course, this comes at the cost of signal to noise, because as you go to the outer edges of the spectra, you really lose in signal intensity. And I'll come back to this practical but really important point, actually. Now, the other point that we have to take into account here is the magnetic moment, which is really highly delocalized in Einthofer clusters. And the way that this is done is through the use of spin projection factors, which effectively take into account how much spin density is delocalized onto your 4 ion subsites. And in principle, we have an individual G tensor for each of these irons, but the way that this is approximated is through a global G tensor. And indeed, we have to think about how this might actually change. We don't know where the G tensor points. So in our simulations, we have to take all these possibilities into account. And lastly, of course, in principle, we have 3 distances, but luckily in the case of Deer, it's basically blind to shorter distances, less than 15 angstroms. So in practice, we only have to worry about this outermost 26 angstrom distance. We have actually tested also that definitely we can ignore these shorter distances, and that is indeed the case. And just to mention here the modulation depth, so you'll see that shortly in the subsequent slides, so how deep your modulation is here, that's really a very sensitive indicator of how many pump spins you're exciting. So that's really a very good check for us to see whether our model is actually correct and whether it's taking into account the right interactions. And last but not least, a major issue for us here is signal to noise, as we are working with 10 microliter samples with less than 10 micromolar, plus the orientation selection that I mentioned, which means you're exciting even fewer spins. So that's a real challenge for sensitivity. And we were lucky to work here in collaboration with John Morton's group at UCL to enhance the signal to noise by more than an order of magnitude by using cryogenic preamplifiers. And actually, this is also something that's going to be coming to Pepper, which I'm very excited about. And that's not unique to Deer. We have indeed actually in this joint paper shown a nice application, I think, to hyperfine spectroscopy with respiratory complex 1. So taking all this, Catherine was able to simulate all the experimental Deer traces, which are shown in black here. And on top, you've got the simulated Deer traces, so the best fit models for A and B. And you can see that clearly the fit is not perfect for model B, but very clearly it must be the correct one because model A doesn't fit at all. And really the great confidence here comes from indeed these outermost field positions here where only the N node and 2 distance can possibly contribute. And because we observe a modulation experimentally, it must be that the N node cluster is the outermost one. Sorry, I realized I've actually lost my N node label here. Apologies for that. And of course, from doing these simulations, we also know actually where the G tensor points, and we know where the spin projection factors are that I mentioned. And that's basically what we got out at the end, what these magnetic parameters are. So having characterized the iron-sulfur cluster EPR signals and assigned them to the structure, we wanted to know, of course, what actually their properties are. And in our case, the first place to look at is the reduction potentials. And for that, again, we could resort to some methodology that we developed for respiratory complex 1, which are these small volume potentiometric titrations, since in our case, we were really extremely limited by the amount of enzyme that we can actually produce. So here we only need about 140 microliters of enzyme for the entire titration, where each sample is only about 10 microliters. Or if you really need to, you can actually push it down to 7 microliters as well. And what you can see here is we're monitoring the potential here as a function of time. So we know that we really are the potential that we think we are at. And these are the corresponding EPR spectra. So I should have probably mentioned that these iron-sulfur clusters become EPR active when they're reduced. So you can see the appearance of, in this case, the N2 cluster. And then we can extract the reduction potential by integrating these EPR signals and plotting them as a function of the potential. And a nice side effect, actually, of these small volume potentiometric titrations is that we can not only do analysis with continuous wave EPR, which is sort of our to-go-to technique as a first characterization, but then we can also do these more detailed post-EPR investigations, such as the DEER that I just mentioned to you, both at X-band frequencies and at Q-band frequencies, which can have different advantages going between different frequencies. So armed with this technique, then Catherine was able to determine the reduction potentials. And what she found, much to our surprise, I have to say, is that N1 and N0, so the two clusters adjacent to each other here, actually have very similar reduction potentials. And the reason why that surprised us is because I think it's somewhat accepted in the field that it's very often what you find in these enzymes with electron transfer relays, and the respiratory enzyme is an extreme with these 8 or 9 clusters, but often you find 3, 4, or even 5, that you have this alternating high and low potential roller coaster energy-like profile. And clearly that's not the case here. And I'll come back to that in my conclusions. We weren't actually able to get a reduction potential for N0 because it's so broad in the continuous wave EPR, but we were at least able to put an upper bound on it from the post-EPR measurements. So what is clear from this is that it's considerably more negative compared to N1 and N2. So this brings me to the conclusions of my first part. So we really started from a blank canvas here, I think, in terms of the biophysics. And what we have at least, I think, made a start on is to place photosynthetic complex 1 on the bioenergetic map of photosynthesis, if you like. And I would argue that it would have been very difficult to do this with any technique other than EPR, combined with lots of challenging biochemistry, of course. And unlike in the respiratory enzyme, N2 here is not the electron sink. And indeed, we have these two adjacent isopotential clusters. And at the moment, we can really only speculate as to what this means. But we have a single electron donor here, as opposed to the 2 electron donor that you have NADH for the respiratory enzyme. So maybe that's needed in order to have these 2 electrons ready for plastoquinone reduction, which of course is a 2 electron process. So there's many questions for us to answer still, of course, also, for example, whether the N0 reduction potential changes when ferredoxin binds, or indeed if we really work under native conditions. So that's something else that we are working on in various aspects in my group to really work in at least artificial membrane systems or in the full membrane systems even. But you have to start somewhere when you have a blank canvas. And I think here the work with the isolated protein was a good starting point. And of course, what we are also interested in, much by analogy with what our mission is for the respiratory enzyme, is to understand what the proton-coupled electron transfer mechanism is. So with that, I'd like to switch gears, and don't worry, it won't be as long as the first part, but I'd like to tell you briefly at least about the method that we are developing here, film electrochemical EPR. So it's been known for many decades, of course, that it's a useful thing to combine EPR spectroscopy and electrochemistry if you're interested in redox processes and radicals. And that's because you have this really nice combination in principle of getting detailed information on structure and bonding together with the reactivity and the kinetics thermodynamics that you can get from the electrochemistry. And of course, you in principle also have the opportunity to capture less intense radicals. But in our case, where we are interested really in redox active proteins, there is a really major limitation with all the setups that are out there. And here on the left-hand side, it's really just a small selection of what's been reported in the literature. And that is when you have a large protein, is that you're just very limited by the diffusion to and from the working electrode. So these are all solution-based setups. And just to maybe say briefly why I think it's important to develop this, well, there's many different redox active enzymes and proteins out there. So if we can really understand how radicals evolve, ideally in real time, whilst these enzymes undergo catalysis with potential control, then I think this would add hopefully some very nice new dimensions here, I guess, in the field of bioinorganic chemistry. So that's our goal. Instead of using solution electrochemistry, so our approach is to combine film electrochemistry, and I'll come back to this, with EPR spectroscopy, so that diffusion is no longer a problem. So can we combine EPR with film electrochemistry to get direct and accurate control of any redox active protein of any size, whilst also investigating the paramagnetic intermediates to gain insight into the mechanism? So very briefly, I'd just like to introduce protein film electrochemistry, because I think not everyone is familiar with this. So the concept of it is actually really simple. So you just basically stick your protein or your redox active molecule onto the working electrode, and therefore you eliminate the problem of diffusion. And in principle means, provided that you can attach the protein in the correct orientation, that you have no limitations on size. Now, there's of course also some challenges associated. Well, the second one here is very much related generally to electrochemistry, is that you don't have the structural information. And that's why protein film electrochemistry has been combined with other spectroscopies. And indeed, in our case here, our wish to combine it with EPR comes with quite a few practical challenges. And this was really all driven by Caltum, who I think has done a terrific job in getting this off the ground. The main difficulty here in combining film electrochemistry and EPR is that film electrochemistry, just like any electrochemistry, of course, requires conductivity, whereas conductive samples are really best avoided in EPR. And since the working electrode has to be inserted into the EPR cavity to enable EPR investigations, this working electrode needs to be tunable, functionalizable, EPR silent, give you high sensitivity, since much of the sample space is taken up by the working electrode. So we, after quite a bit of thinking, I decided that the best place to start would be ITO type electrodes. And actually, this is how we started the collaboration with Erwin Reissler's group in Cambridge, who've developed these hierarchical ITO electrodes. And what Caltum did was quite a lot of hard work, is to translate this to a setup that is usable with our commercial EPR spectrometers. And this is approximately what it looks like here, our PFE EPR setup. And that's just a corresponding SEM image here to show you what this meso-ITO structure, which is actually assembled bottom up from nanoparticles, what that looks like. So we had to first of all benchmark our setup, and for this purpose we chose just about the simplest redox active paramagnet that you might think of, which can be covalently attached to ITO, and that's an amino derivative of tempo. And we demonstrated the immobilization through impedance spectroscopy, as you can see here, and also we can see through the scan rate dependence, the linear dependence on scan rate as opposed to the square root, that we have it directly attached on the surface. And the reduction potential in CV here matches very nicely what's reported in the literature. And what we did to show that we can actually achieve this potential control is to obtain different EPR spectra at different reduction potentials here. I should note that these are frozen solution EPR spectra again, where we have these 3 peaks, because now we have the hyperfine coupling to the nuclear spin of the nitrogen. And we had to freeze the samples because at the time we weren't able to carry this out at room temperature, because we were just too pushed in terms of the high dielectric constant of water combined with the high conductivity of the sample, that we had to go to the frozen state because our Q factor was just not high enough. But now this is actually something that we have overcome since. So again, by effectively integrating the spectra and obtaining the corresponding Nernst plot, we can extract the reduction potential, and much to our relief here, we really got exactly the same value as from the cyclic voltammetry, and that tells us that we have direct and accurate potential control, and that indeed this film electrochemical EPR approach is possible and feasible. And maybe just to mention here that film electrochemical EPR is definitely not there to measure reduction potentials. There's definitely simpler ways out there to do that, such as just recording a CV, or indeed doing potentiometric titrations that I discussed earlier. But this was necessary for us, I think, to demonstrate that we really do have this exquisite potential control. Just as a brief aside, we've also shown an application of this. So this was mainly driven by the Reisner group. And our contribution here was to show that we can directly wire STEMPO, which is another derivative of TEMPO, onto the anode in this case. And the application here was to show some insights into the alcohol oxidation that TEMPO molecules do. And indeed, now we're taking this work further to really figure out what the mechanism of this is with the foam electrochemical setup that we have developed. So that's still work in progress. So we then, of course, had to show the applicability to a protein. So again, we chose something fairly simple, a copper zinc superoxide dismutase, for which the electrochemistry and the EPR is well known. And again, we got excellent potential match here in the cyclic voltammogram and the corresponding film electrochemical EPR titration. So what this showed us is that we have a first example of direct potential control of a buried center in a protein without using any mediators, using our technique, which is really what our first mission was. And just very briefly as an outlook, my current postdoc here, Mariam, is now taking this further to show that we can do it at room temperature in flow with TEMPO. So we can really monitor the signal coming and going as a function of potential. And we've now also applied this to different proteins, such as the MSRP, which is a methionine reductase. Where we are really, I think, onto some interesting mechanistic questions that we are now in the process of answering. So with that, let me just briefly conclude on the part 2. So I think we've shown you that FEEPR is feasible, that we can achieve direct and accurate potential control. And also by using this ITO material, we actually have a very tunable material, and we can tune it for different sized proteins and different types of molecules that we want to look at. Of course, we've got a long way to go. We've actually also shown that it's combinable with pulsed EPR, which I haven't shown you here today. And in the outlook, I showed you that we can actually really do operando studies as well, which we are now extending to catalysis as well. So as an outlook, as a vision, ultimately, I'd like to be able to apply this to really complex proteins, such as the complex ones that I've shown you in the first part of my talk. Clearly, we are still some way away from this, but I hope we are on the right track. So last but not least, let me acknowledge the team of people who made these projects possible for the photosynthetic one work. It's really Catherine who's driven this all, and also other joint members of our team here. This was actually started all off when I was still at Queen Mary before moving to Imperial. And of course, Guy for really getting me interested in the photosynthesis side of things. And thanks to all the other collaborators involved in various aspects of this project. And for part 2, I'd like to thank Erwin and his team for making that possible through providing the STEMPO and also helping us with the ITO materials. And last but not least, thank you so much for listening, and I'll be happy to take some questions later on. Thank you.